Good morning. In this video, we will be studying organic molecules. Of course, you have a very good idea about organic molecules from your study in grade 10 and in grade 11 in biology and human biology. So I think the task would be much easier than for the previous slides. Now, why are we interested in organic molecules? Because all living organisms are actually made up mainly of organic molecules. And as we know, an organic molecule is a molecule that must contain both carbon and hydrogen. And that's why we use and here. So it's not and or, but it is and. So in order for any molecule to be an organic molecule, it should contain carbon and hydrogen. For example, methane gas, CH4, contains both carbon and hydrogen. This is methane. And it doesn't contain any other atoms, but it might contain, an organic molecule might contain carbon and hydrogen plus or minus other molecules. For example, glucose, C6H12O6. It's true that it contains a third element, which is oxygen. However, it has the requirements of an organic molecule, so it has carbon and hydrogen. This might be glucose, or it might be fructose, or it might be galactose, as you know, because they are all structural isomers. But if we come to a molecule like carbon dioxide contains carbon, that's true, but it does not contain hydrogen. So carbon dioxide is considered an inorganic molecule. Similarly, water consists of hydrogen and oxygen. So it has one of the requirements of being organic, which is the presence of hydrogen atoms. But on the other hand, it does not contain carbon. So the water molecule is considered an inorganic molecule. The water molecule is considered an inorganic molecule. Our bodies and our living cells are mostly made of or composed of organic molecules. That's why we are interested in studying organic molecules. Furthermore, most of our food items consist of organic molecules. So we eat carbohydrates, we eat fats, we eat proteins. Inside the human body, or in general, carbon compounds that contain carbon, including, of course, the organic molecules, are very variable. They are numerous and they are different. Why do they have versatile properties? Because of three important reasons. The first one is that the skeleton of the organic molecule that contains carbon is variable. So it might be a linear molecule, or it might be branched, like in case of amino acids, for example, branched, or it might be ring shaped, like for example, glucose molecules in their cyclic form. So carbon skeletons are variable. They might be linear, they might be branched, they might be shaped like a ring. And this, of course, gives them versatility, not only in their structure, but also in their chemical properties and in their functions, because as you know from previous slides, that a different structure of the molecule or different arrangement of atoms would lead to different types of functional groups because of different bonding. Because of this versatility in the skeleton itself, then we can find that we can have a multiplicity of different side chains that can attach to the carbon skeleton. So you might find, for example, a methyl group connected to a carbon atom here, or you might find an amino group, or you might find a hydroxyl group. So the carbon skeletons, as you will see later on, and they are they represent the skeletons of the major molecules of life, can attach different functional groups, and of course every functional group will provide a certain characteristic to the whole molecule. The third variability in the carbon compounds results from the different types of bonding, as we said before, that can form between the carbon atoms. So we can have single bonds or double bonds or triple bonds. And over the coming slides, you will see different examples of carbon atoms that are linked together via single bonds, meaning a single a pair of electrons that is shared, or a double bond where we have like two pairs of electrons that are shared, or a triple bond where the two carbon atoms are connected together via like three pairs of shared electrons. There are four major classes of organic molecules. As you know, we have the carbohydrates, we have the lipids, the proteins, and the nucleic acids. And these four major classes of organic acids are the molecules of life. We will begin by the carbohydrates because they are the most abundant ones in our in the food we intake. And carbohydrates consist mainly of the molecules of carbohydrates consist of carbon, hydrogen, and oxygen. Since they contain carbon and hydrogen, then they are organic. And in addition to this, they contain oxygen. So if you have a molecule whose chemical formula contains only carbon, hydrogen, and oxygen, so it is probably a carbohydrate, but not necessarily a carbohydrate. Because in order for this molecule to be a carbohydrate, there is a condition. And that condition is that the ratio of the hydrogen atoms in the molecule, the number of hydrogen atoms in the molecule to the ratio to oxygen should be 2:1, which is the same ratio of hydrogen and to oxygen in a water molecule. So the general formula of a carbohydrate is actually CH2O, but you can never guarantee that you have only one of this structure, which is H2O. You might have, for example, like in glucose, you might have 6 H2O molecules, or HOH radicals and ions in the molecule, or actually, sorry, atoms and groups. But in this case, the carbon atom, the number of carbon atoms should be the same as that of the number of the water molecules between brackets. So we have C6H2O6, which means if you multiply, if you open the brackets, so you would have C6, 2 by 6 equals 12. So C6H12, and then 1 by 6 makes 6. And so we have C6H12O6. So generally speaking, we can say that the general formula of a monosaccharide is CN, where N is a variable, H2ON. But this applies only to monosaccharides, which are the simplest sugars. But for other more complex carbohydrates, you will find that the formula, the number of carbon atoms, is not equal to the number of water molecules. This is because there is what we call, and what we have studied before, dehydration synthesis, meaning that we remove molecules of water to force the monosaccharides to bind together. So for example, in disaccharides, when you have two sugars, two monosaccharides that bind together, in order for them to bind together, one molecule of water should be removed. We will be seeing the explanation in detail on the following slides, but at this level, we would say that the general formula for a carbohydrate would be Cn, where N denotes the number of carbon atoms, and then between brackets H2OM, and N would be equal to M only in the case of monosaccharides, like we have studied for glucose. By the way, because the chemical structure of glucose involves 6 carbon atoms, and because it is a sugar, so the name will end by O's, which is the suffix for all sugars, and hexa means 6. So all sugars that have the formula C6H12O6 are called hexoses, and these are actually the 3 known monosaccharides in nature, which are glucose, galactose, and fructose. And needless to say that the 3, glucose, galactose, and fructose, are structural isomers. They have the same chemical formula, but a different structural formula. There is another sugar that is also important, and that ends by O's because it is a sugar, but it starts by the prefix penta because it only contains 5 carbon atoms. So because it contains these pentoses, of course, are very important because they include ribose that is present in RNA, and they also include deoxyribose that is present in DNA. So both RNA, so both ribose and deoxyribose are important because they enter in the structure of nucleic acids, but these 2 sugars are considered pentoses. They contain 5 carbon atoms, but since they are monosaccharides or simple sugars, then the formula applies. So if the monosaccharide, the pentose, contains 5 carbon atoms, then it should contain exactly the same number of water molecules. So if we open the brackets and extend the formula, we would have C5, 5 by 2 equals 10. So we would have 10 hydrogen atoms, and 5 by 1 equals 5. So we would have 5 oxygen atoms. So instead of having C6H12O6 as in glucose, galactose, and fructose, we would have C5H10O5. Are carbohydrates important? Yes, of course, because they are very abundant in the human body. We ingest a lot of them, and their bonds store a lot of energy, of chemical energy, and therefore the body breaks down these bonds in respiration to release energy. And 1 gram of carbohydrate, CHO, is is a symbolic formula for carbohydrates. 1 gram of carbohydrates will give you 4 calories if they are burnt in a calorimeter. A calorimeter is a device where you burn carbohydrates and see how much energy is produced. Of course, you all know the dietary sources of carbohydrates, like sugar, like rice, like pasta, like bread, like candies, like cookies. All these products contain a lot of carbohydrates. They give you a lot of energy, and if the surplus of them is stored in the body as fat. And it is important to understand, and we will be going over this later on, that the surplus of carbohydrates is stored in your human body as fat, and that fat is deposited mainly under the skin, and this is what makes you obese when you abuse or when you eat huge amounts of carbohydrates. On the other hand, if you restrict your carbohydrate intake, then to the extent that your body really needs energy and does not find a carbohydrate source, because the carbohydrates are the prime source of energy in your body. So your body will start breaking down fats, and therefore you will be reusing the stored fat under the skin, and therefore you will lose weight again. Carbohydrates include monosaccharides, which are the simplest ones, disaccharides, and polysaccharides. And therefore, if you're ever asked to talk about or to describe the three major classes of carbohydrates, you will be talking about mono, di, and polysaccharides. The monosaccharides are the simplest form of sugars, and the monosaccharides, as we've seen before, the monosaccharides can be, the monosaccharides can be either pentoses or hexoses. But now we're going to go over the three major hexoses: glucose, galactose, and fructose. And as you can see here, in glucose, you have the aldehydic group. In galactose, you still have the same aldehydic group, but you will see that the difference will be in the arrangement of the hydroxyl group here related to carbon atom number 4. 1, 2, 3, 4. So here the OH is to the right, here OH is to the left. The rest of the molecule is similar. And in case of fructose, we have this ketonic group. But the ratio of carbon to hydrogen to oxygen will always be 1 to 2 to 1. So that the ratio hydrogen to oxygen is reversed. Let's take an example, which is, for example, the example of glucose. This is the linear structure of, this is the linear structure of glucose, linear structure or long chain structure of glucose, and this is the cyclic or ring structure of glucose. You will not be asked to draw the structural formula of glucose, but you have to be familiar with it. And actually, when we draw that hexagonal structure, okay, with a CH2OH, this one, CH2OH branch at the end, actually, this is carbon atom number 1, carbon atom number 2, carbon atom number 3, carbon atom number 4, carbon atom number 5, and this is carbon atom number 6. What about this? This is not this is not a carbon atom. This is an oxygen atom. How did this occur? This is an oxygen atom that resulted from a chemical reaction between the two ends of the molecule, this end and that, and after the molecule has been converted into a cyclic structure. On the other hand, the disaccharides will have this molecular formula. Why? Because if you have, for example, glucose, which is C6H12O6, and you add another molecule of glucose, C6H12O6, in order to form a disaccharide or a sugar that is made of two monosaccharides, you need to remove one molecule of water. And this is what we call a condensation reaction. So a condensation reaction is the reaction needed to form a disaccharide, that's to say a sugar that, a bigger sugar that is made of two monosaccharides, or that is made of two simple sugars by allowing them to react together and removing one molecule of water from the medium of the reaction to help them react together. This reaction is called a condensation reaction, but it can also be called dehydration synthesis reaction. Why is it called dehydration synthesis reaction? This is because dehydration means that we remove water and synthesis means making. So we remove one molecule of water in order to synthesize or to make a disaccharide molecule. So if you allow glucose and fructose to undergo dehydration synthesis, they give you the disaccharide sucrose, and it is formed of two simple sugars, as you can see on the slide. If you allow galactose and glucose to react, then you would obtain the disaccharide lactose that consists of two simple sugars. If you allow glucose and glucose to react, then you would obtain the disaccharide maltose. In order for this link to be formed between the molecule of glucose and that of galactose, you should remove one molecule of water, and what remains will be that oxygen that links the two sugars together, and we call this link a glycosidic linkage. So what is a glycosidic linkage? A glycosidic linkage is the link, the bond that forms between two sugars, two monosaccharides actually, during dehydration synthesis. So actually you have a monosaccharide on one side and another monosaccharide on the other side, and they're linked together by an oxygen atom that has a single covalent bond with each of them. What would be the chemical formula of that disaccharide that has been formed upon carrying out dehydration synthesis? Actually, every one of the hexoses that shared in the formation of the disaccharide is C6 or has the formula C6H12O6. So if you add another C6H12O6 and then you remove one molecule of water, then you wouldn't exactly have the double of each of the elements, because there is, in addition, one molecule of water that has been removed. For carbon, it would be C6 plus C6, or C6 by 2 simply, which is C12. Concerning hydrogen, we were expecting the disaccharide to contain 12 and 12, or 24, or double 12. But because there are two hydrogen atoms that have been removed, then we wouldn't have H24, but H22. For oxygen, we are expecting the number of oxygen atoms to be 6 plus 6, or 12. But there is an oxygen atom that has been removed during dehydration, so it is only O11, and therefore the chemical formula is C12H22O11. So this is a wrap-up of the different chemical reactions of condensation or dehydration synthesis that occur in order to obtain a disaccharide. So you have a monosaccharide, C6H12O6, plus another monosaccharide, C6H12O6, and you remove one molecule of water, so you obtain the chemical formula of the disaccharide, which is C12H22O11. Of course, you have to memorize the names of the disaccharides and the individual monosaccharides that form them. So you have to know that maltose is made up of glucose and glucose, sucrose is made up from fructose and glucose, and lactose is made up from glucose and galactose. Of course, maltose is malt sugar, sucrose is cane sugar. So the sugar that we extract from cane sugar is sucrose, and lactose is milk sugar. And that's why milk has a sugary taste even without the addition of any sugar. If the union between, or the chemical reaction between two monosaccharides to give a disaccharide is enhanced by the removal of water or by condensation or dehydration synthesis, then on the other hand, if you want to return the disaccharide to its original state and break it down into two monosaccharides, you have to add this water again. And therefore, the process of hydrolysis is the opposite of dehydration synthesis or condensation. So if you have a disaccharide like this, like sucrose, for example, that essentially consisted of glucose and fructose, but whose molecules have lost a molecule of water and formed this glycosidic linkage between them, then in order to separate them again, you have to return their original water to them. In such a case, water will dissociate into OH and H. One of them will take the H and the other one will take the OH. And in such a case, they wouldn't be forced to bind together and they will separate again into glucose and fructose. And this is simply what we studied in digestion. In order to break down sucrose into its simpler components, which are the monosaccharides, glucose, and fructose, you need water. So as we saw in the digestive system during our study of the digestive system, that hydrolysis is the chemical reaction of digestion of food. It is the chemical reaction that is used in chemical digestion of food. Of course, hydrolysis is the addition of water to break down food, but of course we need enzymes to speed up the process, and the enzyme here would be an enzyme that catalyzes hydrolysis, that catalyzes the process. So it is sucrase enzyme. So what is sucrase enzyme? Because you know that enzymes are specific. So every disaccharide will need a specific disaccharidase to hydrolyze it. So what is sucrase? Sucrase is an enzyme that catalyzes hydrolysis of disaccharides into their components monosaccharides. So sucrase is actually a disaccharidase. It is actually a disaccharidase and not a disaccharide. And you should be very careful when you write down the names because you frequently mix them up. So disaccharidase is an enzyme. And what is a disaccharidase? Or what is sucrose as a major example of disaccharadases? They are enzymes that catalyze the hydrolysis of disaccharides into their component monosaccharides. On the other hand, we have much bigger carbohydrate molecules, which are called the polysaccharides. What are the polysaccharides? The polysaccharides are macromolecules. They are very big organic molecules. They are carbohydrate. Of course, they are carbohydrates, but they are carbohydrate polymers. And here we hear the word polymer maybe for the first time. A polymer is a big molecule that is made of repeated, smaller, simpler units, which are considered their monomers. So monomers react together to form polymers, and polymers break down to form monomers. A polysaccharide should contain at least 3 monomers. So for example, 3 monosaccharides, and they're joined together by dehydration synthesis. In this table, we have several famous examples of polysaccharides. So we have cellulose, and we have to know that cellulose is the major carbohydrate in plants. It is a structural carbohydrate in plants. So cellulose, as you can see, is a plant carbohydrate, and it is a structural carbohydrate, meaning that it enters in the formation, or because it enters in the formation of the cell wall of the plants. And this is at the bottom, you can see how the molecules of glucose are attached to each other, and you can see how the CHOH groups, these ones, are on opposite sides of the molecule, and this is how it looks under the electron microscope. And it is important to know that it is non-branching. It is important here to know that it is non-branching. Now, concerning glycogen, glycogen, on the other hand, is the storage form of carbohydrates in animals. Storage form of carbohydrates in animals. It consists of alpha glucose, and you needn't know the difference between alpha and beta glucose, nor the type of glycosidic linkages, but it is important to know that it is branched, and this is a branching point here, and this is how it looks under the microscope. On the other hand, starch is the storage form of carbohydrates in plants. So the only one that is structural is cellulose. Both types of starch, whether amylose or amylopectin, consist of alpha glucose. Amylose is not branched, as you can see on the diagram, whereas amylopectin is branched. Of course, the more branched the molecule is, the more complex it is. And if it is complex, then the more complex the molecule is and branched, the more difficult it is to be broken down. This is how starch looks like, and you can see that there is branching here. This is the branching point, okay? And this is the O of the glycosidic linkage, and it is a 1,6 linkage because it is a link between carbon atom number 1 and carbon atom number 6. So the branching point forms a glycosidic linkage again, and that glycosidic linkage is a 1,6 linkage because it is between the carbon atom number 1 of one glucose molecule and the carbon atom number 6 of the other glucose molecule. So here we can find a comparison between structural polysaccharides and storage polysaccharides. So structural polysaccharides include only cellulose, and cellulose is found in plants. Storage polysaccharides include starch and glycogen in animals. So in plants, we have structural polysaccharide, or a structural polysaccharide actually, which is cellulose, whereas we have one storage polysaccharide, which is starch. Cellulose makes up plant cell walls, and starch in plants stores excess carbohydrates, as it is in the case of potatoes. Whereas in animals, we have the storage carbohydrate, as we have discussed before, which is glycogen. And glycogen is the storage form of carbohydrates in animals. That's why we call it the animal starch. But I don't like this nomenclature because it makes you confused. It's just the storage form of carbs in animals. Whereas we have another polysaccharide, just like plants have cellulose as being the structural polysaccharides, insects have a structural polysaccharide, which is chitin, and chitin makes up the exoskeleton in insects, as well as the cell wall in mushrooms, and mushrooms, as you know, are fungi. We will stop this video here, and then on the next video, we will be discussing lipids. Thank you.